Could you explain the difference between a lager and an ale?
So the main difference is how they're made and what yeast strain is used to make them.
Are all ales IPAs?
And so a lot of people think pale ale comes from India, but that's not it. A lot of history behind it. So back in the day, the Brits would brew their beer and then would transport it with them when they traveled by ship to India. And it took like several months for them to move from like the UK to India. So what they did was they added a lot of hops to the beer as a preservative to keep the beer longer so it doesn't go bad when they travel for several months to get to India. That's why it's called the Indian Pale Ale.
Mango and pineapple, or they describe different fruit flavor profiles in the beer. Are they mixing those fruits with the beer, or does that come from the variety of hops and different yeast, as you mentioned?
It comes from the variety of hops.
How do you train the yeast to recognize, oh, this is plastic that I need to break down, this is now my new source of food?
Right, that's a good question. So there's a new field called synthetic biology, where you can apply
welcome, Eugene, to the podcast. Thank you so much for doing this. I appreciate it. The other day we had a chat, you and I, in the bar, and funny enough, we were having beers, which is something that yeast makes. So you guys, you're familiar with the way that fermentation works, and I kind of want to get into a bit of the talk about your work, how you engineer yeast to perform different tasks, as well as how you got into science. So I'll kind of let you describe to us what got you interested in science in the first place, and how you found yourself working as a master yeast engineer.
Right, yeah. Thanks for inviting me over to speak on this podcast. So how did I get into science? I'm like, when I was in high school back in the day, I really liked chemistry and biology. Those are two like science topics I always did well in. So I was like, yeah, why not? And then moved to university. Didn't want to do medicine. I wanted to do something closely related to biology and chemistry, but I didn't know what to do. So I was advised to do biochemistry. And so I did. And down the line, my third year of biochemistry, I really loved that course because it blew my mind how every biological process had a chemical basis. That really blew my mind. So it's a nice mix of like biology and chemistry, and that was like what I was interested in. So yeah, I did that, and then really enjoyed the program. And after my undergrad, wanted to do a postgrad, do a master's and a PhD. So I got an opportunity to go to the University of Edinburgh in Scotland to do a PhD in the lab of Chris French, where at the time there was a huge interest in engineering microbes like bacteria to convert wood waste or plant waste into biofuels by synthetic biology and genetic engineering. So yeah, really enjoyed that, and that's how I got into doing academic research. And
So how did you find yourself in Edinburgh? What actually got you to go? Where were you doing your undergrad first, and how did you get the opportunity in Edinburgh?
Right, so I did my undergrad in Ghana. I'm originally from Ghana. It's a nice small country on the Atlantic in West Africa. I did my undergrad there, and at the time when I was about finishing, I had a friend who was doing a PhD in Edinburgh, in Scotland. So she told me about an opportunity to apply for some PhD scholarships over there for overseas students. So I applied for that scholarship, and I was lucky to get in. So it was fun. I just had to show up and not pay anything at all. So I had my tuition paid for. I also was paid a stipend.
That's fantastic.
Yeah, it was amazing. I never had to pay for anything. So I got there, and I think the other main motivation was the lab I was going to work in. So it was a Biotech lab, and they were interested in developing microbial cells for the production of biofuels, and I thought that was pretty interesting. So yeah, that was like the main motivation that got me there.
Then how did you find yourself in Canada here, working with Dr. Myron Smith?
I was like back to Edinburgh. So like, of course, I chose Edinburgh because it's one of the top UK universities. It has a lot of history with a lot of great inventions that happened right there on the campus. So yeah, that really got me excited about that university. And after my PhD in Edinburgh, so I joined a yeast lab in Sweden. And I was lucky to have a very good postdoc advisor and mentor who really got me to enjoy working with yeast. I had no prior experience of working with yeast until I got to that lab. So that project went very well. I really liked it. And then after that, I did a short postdoc at the University of at the Technical University of Denmark in Denmark. And after that short postdoc, I wanted to go back to an English-speaking country. So I was thinking of going back to the UK at the time because I was very familiar with how things were over there. But that was also the time when they had the Brexit thing going on. So they had decided to leave the EU and no one knew how the future of like research funding was going to be. So I looked elsewhere and I found a position at the University of Ottawa for another postdoc in another yeast lab. So I ended up there, did some more work where we tried to find out why yeast were we tried to find out ways by which can make yeast resistant to fermentation inhibitors.
Interesting. Yeah. Okay. We'll get that in a sec. Get there in a second, but I wanna ask you a few things for those that are interested in visiting places, either Denmark or Sweden, or I mean even Edinburgh. During your times there, did you pick up any other languages, like being in Sweden or in Denmark? Did you pick up any other languages?
Yeah, I did. So I learned Swedish for like a year and a half.
Oh, nice.
Yeah, so now I can write and read Swedish like pretty well.
Nice, good for you.
I've got a bit rusty when it comes to speaking, but when I hear people speak Swedish, like, it's all comes back to me. And yeah, and luckily Danish is quite similar to Swedish, but it's more difficult. Yeah, so I'm more comfortable like with Swedish.
Like, could you go into a coffee shop and maybe order a cup of coffee with like a muffin or a donut or something?
Yeah, 100%. Yeah.
Okay, nice. Any places that you recommend visiting if someone is in Denmark or in Sweden or Edinburgh?
Yeah, there's so many places. So it always just in Edinburgh alone, there's like so much to see that you need like multiple days to visit Edinburgh. But it's a very beautiful city. It has like a hill in the middle of the city. So most tourists would go there, climb that hill. It's called an Arthur's Seat.
Okay.
So you climb that hill and it gives you a very nice view of the whole city. So yeah, people do that. And of course, you should see the Edinburgh Castle, like very old castle that's been very well maintained. Also like very in the core of Edinburgh, in the downtown area. And yeah, the other things you can do is so many, if you're into the British drinking culture, there's a lot of like pubs and places you can get very good beer. On like Grassmarket, Haymarket. You can also like walk to most places in Edinburgh. And one fun thing a lot of people also like to see is the Royal Yacht Britannia. So it used to be the yacht that the Queen Elizabeth, like back in the day, would travel around the world in. Yeah. But that yacht got decommissioned and yeah, it's been kept in Edinburgh. So people go there, you go see all the different rooms on the yacht and even the what used to be the bedroom of Queen Elizabeth II is still there. You can just like walk in there and yeah, it's pretty cool to see.
I like the idea of doing a graduate degree in Europe because there's a lot of culture there. And the nice thing about Europe is that you can take a flight from anywhere and go to, for example, if you're in Edinburgh, you can take a flight to Italy or from Spain to Germany, wherever you are. And the flights are fairly inexpensive. You can maybe go for a long weekend, visit a couple of different places, and then come back and continue your studies. It's a very nice place to be in, especially because all the cities are very accessible for with local transportation.
Yeah, that's so true. So you can actually get flights, return tickets to like any part of Europe on those like low budget airlines for like 30 euros or 30 pounds. And exactly right there. So I did that. So it gave me the chance to see like different other countries. So I saw like most of like Western Europe. So I've been to like all the countries in Western Europe because it's that easy to get to all these countries. And they're all like in Europe, they all have like different cultures and different kinds of foods and drinks. That makes it very exciting to just explore.
Good beer, I'm sure.
Good beer, yeah.
What was the best beer you had there?
The best beer, I would say maybe a Czech Pilsner.
Czech Pilsner?
Yeah, so it's a lager that's like very crisp and refreshing.
It's a little hard to avoid now. Could you explain the difference between a lager and an ale?
All right. So the main difference is how they're made and what yeast strain is used to make them. So for ales, the yeast strain is just Saccharomyces cerevisiae, brewer's yeast.
Right.
And these yeasts ferment warm, so they're able to ferment or make ales anywhere between 16 degrees Celsius to 22 degrees Celsius. But for lagers, they are fermented cold, from like 8 degrees Celsius to like 13 degrees Celsius. And with lagers, after the fermentation, so also with the yeast strain, so lager yeasts are different. So they are a hybrid between Saccharomyces cerevisiae, the normal brewer's yeast, and another yeast strain called Eubayanus.
Eubayanus?
Yeah, so it's a hybrid between these two strains. That's what makes it. That's what gives it the cold tolerance feature. Because the Eubayanus is like, it's like very cold resistant, and they actually live under very cold conditions. So they manage to ferment very cold. And so when you make lagers, after the fermentation is done, there's the actual stage called the lagering, where the beer is kept at a low temperature for like several weeks, and that gives it the time for the beer to mature in its true sense, where the yeast would settle out, all the other remaining particles in there will settle out, and it gives it time for the taste also to develop. And that's how you get a lager. But ales are different. So ales are just like fermented warm, and they have a more like hoppy character. It's more like hoppy in there, and the flavor profile is like diverse and richer that you get in a lager.
As opposed to the ale where, so you said the ale is colder, fermented at colder temperatures?
The lagers.
Oh, sorry, lager fermented at colder temperatures.
Yeah.
Ale is fermented at warmer temperatures.
Yeah.
And was it the lager that has a hoppier, more fruity, or different various types of different flavors?
So the lager is more crisp and clean. It says that there's like very little flavors in there. So most of the flavors come from the malt that you use.
Oh, okay.
Or for the ale, that's like richer, it has like more diverse flavors that you get from the yeast and the hops that's added to the fermentation.
Can you explain the malt?
Right, so the malt is the grains that's are used for making the beer. Okay. So for beer, most brewers would use like malted barley, and it's a bit like wheat, but it's a different kind of grain. So what the brewers would do is to mill or to crush the barley, add a lot like warm water to it to extract all the sugars in a process called worting. So they get a very sweet liquid called wort.
Wort, okay.
Yeah, like very rich.
Doesn't sound very appetizing. The wort part doesn't sound.
Yeah, but it's like very rich in sugar, like very sweet.
Yeah.
So so that's what you get from the barley, just the sugars.
Yeah.
And after that, they would add the hops to it, and then it would boil. So it's a bit like making tea. So when it's boiled, all the compounds, the hops, will get like extracted into the wort.
Yeah.
And when that process is done, the wort is like cooled or chilled. Then the yeast is added. The yeast converts the sugars from the barley into alcohol, and then also converts the hop compounds into all the desirable flavors that you get in the beer.
Something I was always curious about is IPAs, which stands for Indian Pale Ale, right?
Yeah.
So where does the name come from? I don't know if it's that obvious, but where does the name come from, and what differs it from like regular ale? Is it a specific type, or are all ales IPAs?
No, well, that's a good question. So a lot of people think India pale ale comes from India, but that's not it.
Okay.
So a lot of history behind it. So back in the day, the Brits would brew their beer and then would transport it with them when they traveled by ship to India, and it took like several months for them to move from like the UK to India. So what they did was they added a lot of hops to the beer to as a preservative, to keep the beer longer so it doesn't go bad when they travel for several months to get to India. That's why it's called the Indian Pale Ale.
Wait, so they added hops to the beer so that it doesn't go bad?
Yeah.
So then how do they make the beer in the first place?
Right. So in making a beer, you add the hops to the worts and you boil.
Right.
And when you boil it, you get a lot of like compounds.
Okay.
And that are extracted from the hops. Some of these compounds are preservatives. They are antimicrobial. So that's what preserves the beer.
Okay.
So in that case, during the stage where they add the hops to the worts, they add a lot of hops so that it gets a lot of those hop compounds. It's like preservatives that makes the beer last longer. So IPAs are usually more bitter than the regular pale ales because of all the extra hops that's been added to the beer.
Okay.
Yeah. But to answer your question, like how different is it from other ales? So it's very hopped. So it's more bitter, and and the flavor, it's also very hoppy. So it's called like very hop forward beer. So it's very hoppy, and it has a very different flavor profile.
So then the distinctive characterization of it is that it's a little more bitter than regular, like than lager beer.
Yeah, a little more bitter, and it's also a little more hoppy. So you get a lot more of the hop character in the beer.
How would you describe hop flavor? Because I tend to stay away from IPAs because I find them a bit more bitter. And I don't really like bitter flavor. But how would you describe a hoppy flavor?
A hoppy flavor is so you get a bit of that skunky that skunkiness in there. So like
delicious.
Hops, yeah, it's like pure hop extract smells like very skunky. Yeah, because all the compounds in there, the thiols and stuff.
Thiols, yeah, okay.
Yeah, so like you get that very skunky flavor in there, but the good kind of skunkiness.
Right. Yes, of course, as one would normally tend to go for. Okay, so then back to fermentation. There are different types of yeast that they use for fermentation, right? And as we've described, it's the main ingredient in beer overall, whether it's lager or ales, is hops, right?
So this is German law. That's like put out like back in the day. This is like beer must be made of hops, yeast, sugar, and barley.
That's it.
Yes, exactly. Yeah. So yeah, so like that's the fundamental or the basic units of like, beer, but it's been modified so many times. So yeah, all the flavor that you get from beer mostly comes from like good interplay between all these different ingredients, like the hops, the yeast strain that you use, even the water profile also makes a difference, and
even the water makes a difference.
Yeah.
So how can water change the flavor? Like, is it the hardness of the water and the different minerals in it?
Yeah, the hardness and minerals in there. Exactly. So some beers you can brew some beers okay with like Guelph water. So Guelph water is very hard. You can't drink it. Like it's a good water source for some beer styles.
Right.
And some other beer styles also prefer water that's not too high in salt. So it also makes a big difference because like the yeast would need some of these minerals or some of the ions and salts for its biological processes. So it contributes to the flavor profile of the beer itself, and also like the function of the yeast metabolism too.
Out of curiosity, do you know how old beer is? Like how early does it date back to any type of evidence that we've noticed people drinking beer?
So it goes way back to the times of the I can't put a number to the years, but
thousands, I imagine.
Thousands, exactly. Yeah, because I mean, like some records of like people drinking beer, like in ancient Egypt, like back in the day.
Nice. Okay.
Yeah, so
I know wine is very ancient as well.
Yeah,
because fermentation isn't a new discovery. It's like thousands of years old. I'm curious to understand the different ways that different beers are made in terms of flavor. Like, how do you enrich a beer in flavor?
Right. So to do that, you need to pair the right strain, the right yeast strain with the right hop variety.
Okay.
So where the flavor comes from is the compounds, the hops. So hop is a very leafy plant material, has all the terpenes and thiols and other chemicals in there. Most of these chemicals are inert. They are not active. They do not have it cannot be perceived as a flavor compound or an aroma compound. So with the yeast coming in there, yeast is able to convert some of these inert or these inactive compounds into more desirable flavors. It's a process called like biotransformation. So for instance, thiol compounds in hops, a lot of hops have a lot of thiols in there, but they are bound to like cysteine or to other molecules in there.
Okay.
So the yeast has an enzyme that will break the bond and then free or liberate these thiol compounds. And it's the free thiols that give beer some of the tropical flavors and aroma that you have in there.
Yeah.
So to get the right flavor profile, you need to pair the right yeast with the right hops to do that conversion.
Are they mixing? So like when they say, oh, this has a fruitier flavor, like mango and pineapple, or they describe different fruit flavor profiles in the beer. Are they mixing those fruits with the beer, or does that come from the variety of hops and different yeast, as you mentioned?
Yeah, it comes from the variety of hops.
So they're not actually mixing fruits in there?
No. Yeah, so it comes from the variety of hops. So some hops, for instance, have a high amount of ethyl called a 3-MHA. So that's a flavor compound in guava. So if you use that hop, so there's one type called one variety called the Cascade. It's actually grows here in Ontario. So if you have like Cascade hops, it has like high amounts of the 3-MHA, which is a guava flavor. So the right kind of yeast, the yeast would biotransform it, so would free the hop, the bound thiols into free thiols, and that's what give the beer the guava or the tropical fruity flavor.
When it comes to like engineering different beer flavors, how do they engineer yeast or the barley to kind of make different flavors? How can you change I guess what genes each can express in order to like change the flavor profile?
Right. So with yeast, there are several ways by which you can do that. So what you can do is you can use a process called hybridization or mating, where you pick one yeast strain that has several copies of the genes that are required to free the thiols, for instance. So you can pick that parental strain, pick another yeast strain that has several copies of another gene that does similar things, and then mate them. Or hybridize them, so you get some hybrid yeast that has the good traits of both parents. So you get this like terrible yeast that can do those like biotransformation more efficiently. So that's one method. You can also use lab evolution, where you continuously grow the yeast cells in wort containing a hop compound like a terpene. And over time, so terpenes, like I mentioned, like these hop compounds are also like antimicrobial.
Right.
So terpenes in high concentrations can kill the yeast cells. So when you grow the cells in worts with the terpenes over time, the yeast will adapt a way to detoxify it by converting the terpenes into something less toxic, which in most cases is another terpene that has another flavor profile. So that could be another option. And the third option that's like very tricky is the genetic engineering that brings up a whole lot of controversy, because it's going to use a yeast for a food application. So you can also pick, engineer the cells to express an enzyme that would free the bound thiols to release all the flavor compounds in the hops. How
do you determine whether or not a strain is like a good candidate for fermentation to use as a good source for a certain flavor?
Just
trial and error.
Yeah, screening. So you screen, and for like yeast labs or for brewers, what we look for is how well they ferment the sugars in the barley, because that determines how long your fermentation process is going to take. And in the world of brewing, like time is money, so a lot of brewers would prefer a yeast strain that will ferment in a short period of time instead of like a longer time. So let's say it takes you 14 days to ferment or to make beer. You'd want a yeast that can do that same thing in 7 days. That way in that 14 days, you could have done like 2 batches of beer already. So like how fast it ferments the sugars, and also how it's able to transform the hop compounds into desirable flavors in the beer. That's another thing to look for. A third trait that people are interested in is how well they flocculate, or how well the yeast like fall out of the beer after fermentation, because some beers are sort of brewed such that it's like very crystal clear, you can see through it, and so you need a yeast strain that would do the fermentation and it would fall out of solution, and then you get a very clear part of the beer, which you can sell. In that case, you don't need to really filter it a lot, because that could also be very costly.
So then in terms of modifying the process of fermentation, can we do that in order to make yeast more efficient?
Yeah, we can. To increase fermentation, or the rate of fermentation, what happens is the cells would chew through the sugars in the wort. And one way of doing it is to get the sugars into the cell in the first place. And this process is mediated by transporters. So there are some sugar transporters that get the sugars into the cell. So if you want to increase the rate of fermentation, you can engineer the cells to express or to produce more of these transporters that will get the sugar into the cell. And when it gets into the cell, you can also engineer the cell to make all the enzymes or the tiny bits of proteins in the cell that chews or breaks the sugar into alcohol, into energy. So expressing those enzymes as well would increase the rates of fermentation.
Could you describe the process of fermentation? How does it work, and how does yeast ferment sugars in order to produce alcohol?
All right. So firstly, taking the sugar, and then the sugar is broken down through a series of reactions called glycolysis into pyruvate. And then this pyruvate is now converted to alcohol or ethanol. So that's basically the process of fermentation. That's what happened.
It's a single cell organism, right?
It is.
How do they replicate? And they survive by eating sugar, right?
Yeah.
So how does yeast replicate? Can you tell us a bit more about it?
So there are two ways. It can do that sexually by forming spores.
Okay.
And then the spores then germinate under the right conditions to make like more yeast cells. Yeah. Because they also do that by just like biting off each other. So the cells will just split into two, then two splits into four, four into 8, and that's the most common way by which they divide or grow.
Okay. And then what did you learn from your work with yeast in Sweden? What was it, Sweden?
Yeah, Sweden.
Sweden, yeah.
Oh, yeah.
What were you guys working on at the time?
Yeah, it was for different applications. So at the time we had a collaboration with Samsung, the mobile phone company, to produce lactic acid from fermentation. And the reason why they were interested in lactic acid was they wanted to make a bioplastic called PLA, polylactic acid, for their mobile phones.
Interesting. What was the plastic used for?
To make the cover of the mobile phone. So it's like a bioplastic.
Right.
Yeah.
Just to make it a bit more environmentally friendly.
Exactly.
Okay.
Yeah. But then lactic acid, I mean, it's an acid, so it's high concentrations that kills the cells. So our work was to engineer yeast cells to become more resistant to the toxic effect or the lactic acid. And we did that. We did lab evolution where we forced the cells to grow under very acidic conditions. And we identified some gene mutations that could be targeted to make the cells more resistant to the toxic effect of lactic acid.
Are there challenges that brewers have in terms of growing their hops or growing the barley that can be improved by modifying the gene? I mean, I guess I'm asking about genetic modification here, about genetically modifying the hops or barley in order to have either larger production volumes or just better flavor profiles.
So in terms of flavor profiles, so the brewers don't do that job. They don't do the engineering. They don't engineer the hops or the barley. They just like buy them and use it. So improve the flavor profile mostly comes from the yeast itself. So that's where a lot of the engineering goes into. And so I help yeast companies or academic labs that engineer yeast to effectively convert the hop compounds into useful or flavors and aromas. And in the world of the hops, so there are some hop growers that also do plant breeding, where they take different varieties of hops to breed them to get a hybrid that has high amounts of terpenes or thiols that could be used to make beer. And in that sense, so beyond that, the brewers get very excited because like hops is expensive. So if you have a hop variety that has a high amount of the terpenes or the hop compounds, you'd rather want to go buy those hops instead of the traditional ones that have like low amounts of terpenes or hop compounds.
What's your current project right now? What are you currently working on?
Right here at Carleton, I'm trying to use yeast cells to convert plastic waste into useful chemicals. So what a lot of people are doing now is there's a lot of work going on into breaking down plastic waste, like PET plastics, like you find in bottles, plastic bottles. You're using like enzymes or like chemical reactions to break it down to its most basic units or the fundamental units. And that's it. For us, in my research lab, what we're doing is we're trying to engineer yeast to recognize these broken down products or these basic units of plastic as food. So they can consume it as a carbon source and then ferment it to produce ethanol or biofuels that can be used for cars and engines.
So that's very interesting, because you can take yeast and now give it a completely new function other than making beer or growing or making bread. How do you train the yeast to recognize, oh, this is plastic that I need to break down. This is now my new source of food?
Right, that's a good question. So there's a new field called synthetic biology, where you can apply some genetic engineering tools to train cells to do something that doesn't do normally.
Right.
So to do that, in the yeast cell, there's a DNA that has all the instructions that tells the cells what it should do. So when the yeast cell like comes into contact with sugar, the DNA would make enzymes that would break down the sugar to produce biofuels. So with synthetic biology, you can bring in a foreign DNA that can bring in a foreign DNA, put in a yeast cell, and then it trains the cell to recognize these plastic compounds as food. And then through a series of biochemical reactions that are mediated by the enzymes that the DNA would make, the enzymes would transform the plastic compounds to biofuels through a series of steps.
Okay, so we're not only breaking down plastic, you're actually also making a product from it that we can use for something else.
Exactly. And about like training cells, so there are some bacteria that does this naturally. So they grow in refuse dump sites. The only thing is that these bacteria are difficult to grow.
Okay.
And this will require very strict nutrition and very strict growth conditions to break down the plastics. So essentially what we're doing is we're looking at the genetic databases of these bacteria, and then we'll pick up the instructions that the cell uses to recognize plastics as food and copy these instructions, put that into yeast cells. And then now the yeast gets trained to read these instructions, and it's able to recognize plastic waste as food and would convert that into biofuels and other chemicals. That's the approach that we're using right now.
And is the biofuels produced by yeast? Does it is it any different than the biofuels produced in any other methods?
No, it's all ethanol, bioethanol. Yeah, the only difference with the other methods is like the feedstock that is used. Some people would make biofuels from like corn or from sugarcane, but the final product, which is the bioethanol, is like the same thing. It's the same chemical, really.
Right. Yeah, it's the same chemical. Okay.
It's just the process that's different.
Just the process that's different. Yeah. So then, why wouldn't we how many different types of plastics can we actually use to train yeast at breaking down these plastics into biofuels?
So we don't know yet. So this field is a little like very new.
Yeah.
There's still like a lot of work that needs to be done. Because now there are enzymes that will break down plastics to the basic units, but they are not too efficient, to be honest. So there's a lot of work going on to improve these enzymes, to make them more efficient and to increase the activity. So yeah, we don't know yet. So it's still like a lot of work that needs to be done in this field.
Any idea of how long it would take to break down one bottle of plastic, for example, using yeast?
So there's a company in France that has a modified version of the enzyme that breaks down PET plastics. And apparently, the most efficient enzyme that they have now takes about 60 days to break down plastic, which is like very slow to break down.
Yeah,
that's
a long time.
Yeah,
to break down just one bottle of plastic.
I'm not too sure like the quantity
Okay.
yet, but yeah, apparently it takes that amount of time to break down PET plastics.
Man, so clearly one of the reasons why we should start moving away from plastics, but that is also promising that there's something that can break down plastics a bit faster. Is it possible to engineer these, to actually genetically modify these different strains of yeast to break them down faster? Maybe produce more enzyme or produce a stronger reactive form of that enzyme to break down the plastic?
Yeah, 100%. So right now, what we need to do is to make to produce like more efficient enzymes. And people are trying to use like different tools like AI technologies to predict what combinations of amino acids to make. They're using like AI tools to really enhance or to make better predictions of how the most ideal enzyme should be,
right,
to increase its activity or its like rate of breaking down plastics. So mostly being able to increase activity and also be able to produce a lot of the enzymes. I think that's what we need to work on right now.
Okay. Have you used AI at all? Have you used AI to try to modify anything in the lab or to make a process more efficient?
Not at the moment, because for the kind of work that I need AI for, I don't think we have enough data to train our models to do what we want it to do. But eventually, I think that's going to happen in my group.
Okay, awesome.
Yeah.
What kind of yeast have you found to be best at breaking down these plastics?
So yeast don't do this naturally. So what we're trying to do is to engineer them to do that.
And what's the best candidate model for engineering? Can you use any yeast for that? Like even the one that we use to make bread?
Yeah, you can use that. So you can use
Really?
the baker's yeast, or like brewer's yeast. Yeah.
So you don't actually have to have some prerequisite gene structure or profile in order for you to train them or to engineer them to break down plastics.
What you need to do is find a yeast strain that's more efficient in producing the enzymes, and that is a limitation with the baker's yeast right now.
Okay.
So people would rather use other strains of yeast called like Pichia because it's able to produce enzymes or release enzymes more efficiently, or aspergillus. But that's a different kind of fungi though. But yeah, Pichia in terms of yeast, yes. Or with a baker's yeast, you can do that, but they do not efficiently express or produce these enzymes. And there's still a lot of work going on to make them more efficient in protein production.
What's the purpose of that enzyme aside from us discovering that it can break down plastic? What's the natural function for that enzyme in yeast?
It has to function in yeast. So these enzymes are mostly produced by bacteria.
Oh,
so say like you have to engineer yeast to make them.
Gotcha.
So
okay,
yeah, yeast don't naturally make them.
Which is why. Oh, I see what.
That's why you have to engineer them to produce it.
Yeah. So you need to find a way to, because you can take that bacteria and or you can take the gene from that bacteria that produces that enzyme and put it into any type of yeast.
Exactly.
But then depending on the type of yeast, it will make it either more or less efficient.
Exactly.
Gotcha.
Yeah, so it's a background strain that really makes a difference.
Okay. Yeah. You're also a professor here at Carleton, right?
Yes, I am.
Yeah. What are you teaching right now?
So I teach a 4th year biotechnology class. It's called like current topics in biotechnology.
Yeah.
It's a fun class, a small class. So what we do is we pick wide aspects of biotechnology each week, and then we discuss like new technologies that are being developed by different companies to solve a problem. And we get the students to think through these problems, to think through the technologies or the science behind the technologies, and to think of some other challenges that they might face in terms of like ethical considerations or like increasing public perception, and also even with like regulations and stuff. And with all these put together, the students will be coached to start their own to create their own biotech startup, and develop their own technology to solve a problem using biotechnology. So it's a fun class, and it's like very interactive, nice to discuss like all the new trends that are happening in the world of biotechnology.
That's very cool. Have you encountered any students that used yeast to start a company of their own?
Not yet.
In the course?
Not yet. I'm trying to like push them to think about using yeast, but
okay, this is a good product to be using.
Yeah.
What's one of the current projects that the students are working on that, or like the companies that they're making right now, that is as close to being achievable as possible?
So right now we're just like halfway through the course, and they're still like developing the idea, so it's not really concrete right now.
Right.
But I think one group has a cool idea of producing enzymes like microbial cells that can clean up oil spills in the attic. Which I think is pretty interesting, but yeah, we're still trying to shape the idea a little bit.
Yeah, it's very interesting because I've read a little bit about bacteria that can do that, right?
Yeah.
But can yeast do that? Can you take yeast, engineer it to, because you can do the same thing with bacteria where you can take part of its genome and embed it into yeast in order to break down plastics, and do the same for yeast to be able to break down biofuel or just fuels in general, like fossil fuels?
Yeah, exactly. Oil. You can. So there's some bacteria that does its job naturally. So you can take some of their genes and then stick into yeast using synthetic biology tools, and that way you can train yeast to do the same job. So it's possible. Yeah, it can be done.
And what's your future hope for your current research?
Well,
or your aspirations? Where do you see your research going in the next 5 to 10 years?
The next 5 to 10 years, I like us to have like a fantastic yeast strain that can consume plastic compounds and convert them into like biofuels or to other biochemicals in a tank. So my big dream is how like work with companies or industries that are into waste remediation, where they could have like a fermentation process where they have a tank just like you'd find in a brewery. Have a tank with like hydrolyzed plastics in there with some yeast strains in there. The yeast would chew through the hydrolyzed plastics and then convert it to biofuels or to other interesting biochemicals.
That'd be awesome if you could also like make a machine. You just dump the plastic into it, and then you get fuel out of it that you can use it for something else.
Exactly. Yeah.
Or just ethanol to just make yourself some ethanol from dumping plastics.
Yeah, ethanol. Even like hand sanitizers, which are also like ethanol.
Oh, true. Yeah.
So, yeah, to be a fun project, to have like a small like a home
System,
like
yeah.
a system like a coffee maker. You put in your waste plastic, you crash it, you add a sachet of yeast to it, ferment it to make ethanol that can be so like used to make like hand sanitizers or something. Yeah,
That'd be pretty good, because I remember taking a industrial biochemistry course once in undergrad, and there's always like what companies have in mind is always this process of, if we're making a product, what can we do with the byproducts of it, because they want to make it as least wasteful as possible. So you can like take a certain product as you're breaking it down and maybe make soap out of it, or make other ingredients out of it that you can use as separate forms of income, as opposed to just dumping it and throwing it out.
Exactly. So that's what we are interested in. So with PET plastics, for instance, so PET is like a long chain of like smaller units called terephthalate acid and ethylene glycol. So long chains of these music units. So we break down PET plastics to get these individual compounds. So ethylene glycol is what is used as an antifreeze agent.
Right. Yeah.
That's like used in cars. But for the other compound, the terephthalate acid, there's no use for it right now. That's like a waste product of a waste product.
Right.
So our plan is to engineer yeast to recognize TPA or terephthalate acid as a carbon source and ferment it to produce something useful. So yeah.
The molecule itself, is it possible to be broken down currently? Is it possible to be broken down into anything that would be useful? Is there any structure within it that would be useful for some other product? I imagine that if let's say you take a third of it and that becomes useful, but then there's still 2/3 that you have nothing to do with, but at least something.
At the moment, I'm not too sure if it has any use as it is. So it's a ring structure with 2 acidic groups on it.
Okay.
So
what are the acidic groups? Do you know?
Just carboxylic acid, so COOH.
Okay.
Yeah. So in terms of like breaking down to something useful, I can't think of anything right now. No.
I don't know if like you can remove one of the groups and it just becomes benzenoic acid, and then
possibly, yeah,
might be able to use it in as a reagent in processes, because interesting stuff.
Yeah.
Man, I really appreciate you coming on the episode. Thank you so much. I know a little bit more about beer now. My cousin really likes beer, so I could use this knowledge now to have discussions with him about beer. But thank you so much for coming on the episode. I really appreciate your time, and I wish you all the best with your work, your research, and your students, and I hope to see you soon in the future.
All right, thanks George, thanks for having me on your podcast.
Thank you so much, man. Take care. Have a nice day.
You too.
Thanks for listening, everyone.